Okay, dear students, we will start our lecture series of physiology. Lecture number 21 is on reproduction and endocrine glands. We are going to start new chapter here, reproduction and endocrine glands. Reproduction means again production or again creation. Re means again, production means production or creation. And endocrine glands are the glands which are secreted by the cells directly into the blood vessel without any duct. Therefore, it is called as endocrine. So these are secreting from the internal side. So here we will start the sixth chapter, and in this sixth chapter we have to study the physiology of male reproduction, in which hormonal control of spermatogenesis, physiology of female reproduction, hormonal control of menstrual cycle, structure and function of primary pituitary, thyroid, parathyroid, pancreas and adrenal glands. We have to complete this syllabus of unit number 6 in 8 lectures. So 6.1, first topic of the chapter number 6 is physiology of male reproduction. Generally, in male, the reproductive organs are testes, and these testes are located in the scroll sacs of the male sex organ, and these testes have a series of ducts and glands, and which together form a male reproductive organ. So sperms are produced in the testes and are transported through the reproductive ducts. These ducts include the epididymis, vasa deferens, ejaculatory duct and urethra. These are all the organs we will see in the next figure. The reproductive glands produce secretions that become part of semen, or we can say sex fluid, the fluid that is ejaculated from the urethra. These glands include the seminal vesicle, prostate gland and bulbourethral glands. So testes produce sperms and they enter in epididymis, and epididymis, which is the long tube, coiled tube that rests on the backside of each testicle, and it transports and stores sperm cells until the maturity. The epididymis opens into vas deferens. The vas deferens is a long muscular tube, and vas deferens transports mature sperm to the urethra. Then from here, ejaculatory duct is formed by the fusion of this vas deferens and duct of seminal vesicles, and both together unite to form a next tube, urethra, and they pass through the prostate and opens into urethra. So the way is through the prostate gland, where the prostate secretion is added for lubrication or nourishment. Urethra is a tubular structure that transports semen and urine. And in this way, all the structure like epididymis then distributes and up to the urethra. So vas deferens extends and continues up to the urethra for its secretion or seminal fluid secretion, we can say. So this is the basic structure that we will discuss here. The testis, this is the testis, which is enclosed in an outer membrane called as, or we can say this whole structure is called as scrotum, in which this testis is located, and this testis is enclosed in a tunica albuginea from the outside and tunia vaginalis from the inside. we can say this is the structure, or we can say this is the outside and this is the inside structure. If we think that this is the posterior part, then the tubule, the spermatic cord enters from here, or we can say the semen formed is ejaculated from this tubule, one and the same. This is the vast difference tubule. When it becomes coiled, then it is called as epididymis in the separate chamber, and then whenever it enlarges and enters with a random scattering form, it is rete testis. This is the rete testis area. Then it forms a lobules. These are 1, 2, 3, 4, 5 or 6 lobules, and in each lobule there is a tubule called as seminiferous tubule. Seminiferous tubule is the original tube, or we can say main tube, in which the sperms are produced. So this is the structure of the testes. Now we will see the histological structure of testes. What kind of cells are there? The testes have an unusually thick connective tissue capsule called as tunica albuginea. An unusually thick dense connective tissue capsule, the tunica albuginea, covers each testis. The inner part of this capsule, the tunica vasculosa, is a loose connective tissue layer that contains blood vessels for support and supply of nourishment, oxygen, etc. Each testis is divided into approximately 250 lobules by incomplete connective tissue septa. There is no separate membrane, and that project from the capsule. Along the posterior surface of the testis, the tunica albuginea thickens and projects inward as the mediastinum testis, blood vessels, lymphatic vessels, and the genital excurrent ducts that pass through the mediastinum as they enter or leave the testes. Each lobule consists of several highly convoluted seminiferous tubules, which are the basic unit where the actual sperms are developed or formed. Each lobule of the testes consists of 1 to 4 seminiferous tubules in which sperm are produced, and a connective tissue stroma in which Leydig or interstitial cells are contained. Each tubule within the lobule forms a loop, and because of its considerable length is highly convoluted, actually folding on itself within the lobule. The ends of the loop are located near the mediastinum of the testis, where they assume a short straight course, and this part of the seminiferous tubule is called the straight tubule, or we can say tubulus erectus. It becomes continuous with the rete testis and anastomosing canal system within the mediastinum, because rete testis is a short structure in mediastinum where the random and anastomosing canal system through which the sperms are passes. Now we will see the microscopic structure of seminiferous tubules in which the sperms are produced. Actually, there are two types of cells found in testes: Sertoli cells and spermatogenesis cells, or spermatogenic cells, from where the actual sperms are developed. And Sertoli cells are the supporting cells or nourishing cells which give nourishment to the cells. The seminiferous tubules consist of seminiferous epithelium, which is responsible for the formation of germ cells and germ cells, that is spermatid, and then from spermatid the sperms are developed, are surrounded by a tunica propria. Each seminiferous tubule is approximately 50 centimeter long. Actual range is 30 to 80 centimeter, but it occurs about 50 centimeter long and 150 to 250 micrometer in diameter. So the tubule is about 150 to 250 micrometer in diameter. The seminiferous epithelium is an unusual and complex stratified epithelium composed of two basic cell populations, that is Sertoli cells, supporting cells, and spermatogenic cells, sperm producing cells. So first of all, we will see what are the Sertoli cells. Sertoli cells are embedded among the spermatogenic cells in the seminiferous tubules and are large Sertoli cells, or also called as sustentacular cells, or also called as supporting cells, which extend from the basement membrane to the lumen of the tubule. We will see figure 6.1. This is figure 6.1, in which testes A, B and C parts are shown. In A part, interstitial cells of the testes are shown. These are the interstitial cells which are located in between. This is the tubule, this is another tubule, and this is another tubule. These are the seminiferous tubules which are circular in structure, and in between these two tubules interstitial cells or Leydig cells are present. Some fibroblasts are also there, and blood vessels are common for support of nourishment. Then in second, cross section of seminiferous tubule. This is the actual cross section of seminiferous tubule where the sperm or semen is produced. And in third, these are the sperms which are produced, these tails and sperms. Interstitial cells, these we have seen, these interstitial cells are here. So this structure is magnified here, and this structure is magnified here. This means the this is the tunica albuginea layer. Cells of Sertoli, these are the Sertoli cells, supporting cells, random, and these are the spermatogonial cells developing into the sperm at last. So from spermatogonium cell, first, second primary spermatocyte is formed, third secondary spermatocyte is formed, and fourth spermatids are formed, and finally the spermatozoa are developed, mature spermatozoa. This is the structure of seminiferous tubule and lumen. 6.1. These cells do not replicate after puberty. Sertoli cells are columnar cells with extensive apical and lateral processes that surround the adjacent spermatogonic cells and occupy the spaces between them. Sertoli cells give structural organization to the tubules as they extend through the full thickness of the seminiferous epithelium. Sertoli cells support and protect developing spermatogenic cells in several ways. They nourish spermatocytes, spermatids, and sperm. They also produce fluid for sperm transport, secrete the hormone inhibin, and regulate the effects of testosterone and FSH or follicle stimulating hormone. Second is spermatogenic cells. Stem cells called spermatogonia, which regularly replicate and differentiate into mature sperm. These cells are derived from primordial germ cells originating in the yolk sac that colonizes the gonadal ridges during early development of the testes. Spermatogenic cells are organized in poorly defined layers of progressive development between adjacent sertoli cells. These are the sertoli cells and these are the adjacent cells. Or we can say in this whole seminiferous tubules there are sertoli cells randomly arranged, or at least 4 are there from the different 4 sides. They are not many in numbers, they are in limited numbers. So the most immature spermatogenic cells called as spermatogonia rest on the basal lamina. This is the basal lamina where spermatogonium is resting. Then the spermatogonia, in order of advancing maturity, or we can say development, they are primary spermatocytes. They develop into primary spermatocyte, then they again develop into secondary spermatocyte, and then they form spermatids. These are the spermatids, and the most mature cell called as spermatid or sperm cells, or we can say spermatozoa finally. And these sperms are attached to the apical portion of the sertoli cells for attachment and support, where they border the lumen of the tubule. And in this way, the seminiferous tubules forming the sperm cells and supporting them and nourishing them. Spermatogenesis. The process of formation of sperm is known as spermatogenesis, or we can say the development of spermatozoa takes place in the male gonad called as testes, which are paired in structure. It's called as spermatogenesis. Simply another way, in mammals they are suspended in scrotal sac or scroll sac outside the abdominal cavity. The testes consisting of seminiferous tubules which remain enclosed within connective tissue and separated from each other by interstitial cells. Means in between these tubules, the interstitial, in between the seminiferous tubules, interstitial cells are there. The cells secrete male sex hormones called as androgens. The seminiferous tubules are composed of both the germinal, that is reproductive, and somatic, that is supporting or body cells. The germinal tissues lines the seminiferous tubules as a germinal epithelium which undergoes a process of spermatogenesis. Means actual spermatogenesis occurs on the germinal epithelium to form spermatozoa. Means the process of formation of sperm from spermatogonia to spermatocytes to spermatids to spermatophores. This is the process of formation of sperm. And the presence of somatic cell in the seminiferous tubules is a special feature of vertebrate testes called as sertoli cell. That we have already seen. The sertoli cells are the supporting cells among all these sperm producing cells. Now, the spermatogenesis is a continuous process in which the sperms are formed, and it is divided into 2 steps. One, formation of spermatids. The cells of germinal epithelium, when produced spermatozoa, are called primordial germ cells. It undergoes the following phases, that is 3 phases: phase of multiplication, phase of growth, and phase of maturation. This is the 3rd phase. So we will see one by one. First is the phase of maturation, in which the spermatids going to multiply phase of multiplication. They multiply, then they grow and they mature. This is the step of formation of sperm. The undifferentiated primordial germ cells contains large sized and chromatin rich nucleus. It undergoes repeated mitotic cell division and become sperm mother cells or spermatogonia. Each cell is 2n number of chromosomes, that is diploid chromosome, and enters into the phase of growth. Second phase of growth. During this phase, the spermatogonial cells accumulate large amount of nutritive material or nutrition and chromatin material. Their volume becomes double and each cell is called as primary spermatocyte. This primary spermatocyte is also diploid in nature, means having 2n number of chromosomes. And the third phase is the phase of maturation, where the multiplied and grown cells becomes mature, that we will see in the next lecture. Till then, see you.